One of the most important groups of nematodes globally are the cyst nematodes. We will use the soybean cyst nematode as an example of this group. There are other cyst species that attack potatoes, wheat, vegetable crops, including sugar beets and table beans. The soybean cyst is a pretty important story and pathogen in the United States. It is arguably the most important pathogen of soybeans in the U.S. It was first found in North Carolina at an agricultural experiment station near Castle Hayne in 1954. And it was originally believed that it was introduced on soybean seed as part of a plant breeding program at that station. It was subsequently determined that it was probably actually brought in with soil from Asia that had been brought to the U.S. many, many times in many locations for work on rhizobium bacteria and spread throughout the U.S. This development probably was why initial attempts at quarantine of this pathogen in the North Carolina region was never effective. Soybeans were a minor crop in the 1950s, became a major crop in the 1970s, and every time they looked at a new soybean field, they kept finding the pest as soon as they started planting soybeans and seeing them. Thus, it was moving too fast to be accounted for by movement of the pathogen then. And it was probably because these fields had been contaminated earlier, it survived on weeds and from the rhizobium research, and then when we started planting lots of soybeans, we found lots of the pathogens. This map shows the distribution of the soybean cyst nematodes in the United States. This is from the late 1990s. And by this time, nearly every soybean producing county in the United States is infested with the nematode. It's called the cyst nematode because at maturity, the adult female forms this protective sac called a cyst. The infective stage is the second stage juvenile. It is the only infective stage for this nematode, is the second stage juvenile. But at maturity, it produces this globular female. So it grows from this stage to this globular, sort of pear-like, lemon-like shape as a mature reproductive female. And then upon senescence, this white female, and these are many females on one soybean root, undergoes a tanning process. The cuticle becomes very dark, becomes leathery, and inside are numerous eggs. And this is a protective structure for survival. And we look at the life cycle of this particular nematode. We start with a cyst that's in the soil, this protective female. It'll fall off the roots after the crop senesces and overwinter in the soil. In the springs, the juveniles begin to hatch. They invade the root tip, normally near the cortical regions. And as again, these are the only infected stage, unlike the lesion nematode, where all stages are infected. For the cyst nematodes, only the juveniles are infected. They migrate to the roots to the pole of the vascular tissue opposite the xylem or the phloem. They begin feeding on pericycle and endodermal cells and induce the formation of these specialized nurse cells that which we call syncytia. The syncytia developed as from the coalescence of adjacent cells to form individually large, highly metabolic active cells that provide all the nutrition needed for the development of this nematode from this little worm through the juvenile, advanced juvenile sausage-shaped stages, eventually to the swollen female. An interesting thing about this one is that the males and females are identical until the last molt. The female becomes a sort of pear-like, lemon-like structure, whereas the males revert back to the worm-like structure. And then it is mobile again in the soil, crawling around to find the females for mating. One interesting and difficult component of the cyst nematode is the absence of diagnostic symptoms during early stages and minor levels of infestation. However, you can still be experiencing yield loss. Some research done by Terry Neblak at the University of Illinois clearly demonstrated in this soybean field where the plants just looked gorgeous, great looking crop of soybeans, but it was infested with the cyst nematode, and where the field was treated with a nematicide, there was a 9 bushel per acre yield increase relative to the untreated portion of the field. So unless they're treated with nematicides, which is too expensive for this crop, it is very difficult for a grower to tell that he's losing yield due to this pest. Usually management of the cyst nematode is a combination of host resistance and crop rotation. Nematicides are generally not used, too costly, and because of environmental regulations today, there are very few that are labeled for use on soybeans. The soybean cyst nematode, unlike the lesion nematode and the root knot nematodes we'll talk about in a future lecture, have a very narrow host range. Soybean and common snap beans are about the only 2 crops that are susceptible in the United States, plus a few weed species. So a rotation to a non-host, such as corn, wheat, potatoes, is an effective way of reducing the population. So a management system that has some use of host resistance and the use of rotation to a non-host crop will effectively suppress the population and avoid serious yield losses. Resistance itself can be compromised because we have relatively few resistance genes and we have multiple pathogen races that compromise that resistance. This slide illustrates the value of an effective rotation to a non-host crop. In this soybean field, this was in North Carolina, this half of the field was rotated with corn, and you see a nice healthy looking soybean crop. This half of the field had continuous soybeans, and over a 5 or 6 years period of continuous soybeans, the population density in this soil was so high that there was major damage, the stunting, fluorosis, and essentially no yield in that portion of the field. A dilemma when dealing with soil-borne pathogens, and nematodes in particular, is that of applying a management tactic to an entire field when only a portion of the field is infested. With most pests such as this, they rarely infest the entire field. You tend to find hot spots where you can have severe damage. But management is applied to the entire field. So a grower has to consider the profitability of applying a management tactic that's going to raise his production cost to an entire field when the problem is limited to portions of that field. The last concept to manage, to talk about with cyst nematodes is that of movement and the role of human activity of movement of these pests, pathogens around the world. And this is typical of many plant pathogens. In this case, we'll talk about the potato cyst nematodes. There are 2 different species of potato cyst nematodes. They developed in the Andes region of South America where the potatoes originated. The Spanish took potatoes to Europe. When they did, they took the pathogen with them. In the early 1700s, the potato became widespread in Europe as a food crop, especially for the peasants. And pretty soon, the potato cyst nematode was throughout all of Europe. And it wasn't distributed to the United States until 1919. It wasn't discovered in the United States until 1940. And it turns out that in 1940, a farmer on Long Island was having a problem with the potato crop. He called in scientists from Cornell University. They diagnosed it as potato cyst nematode. His farm was a point at which soldiers coming back from Europe at the end of World War I disembarked. In a major camp there, tents, equipment from Europe, The battlefields in Europe were old potato fields, and when they came back, they brought soil from the potato fields as contamination and the cyst nematodes as contamination. And it took 20 years for the population of cyst on the potato farm in Long Island to increase enough to yield symptoms. Shortly thereafter, they quarantined Long Island to prevent spread of the cyst outside of Long Island. Unfortunately, the grower on the farm it was first found sold that farm and moved to upstate New York, started a new potato farm in Steuben County. 20 years later, this pathogen was found on that farm. Shortly thereafter, the U.S. government came in and bought the farm and turned it into a quarantine research farm. And that was pretty much all the cyst spread in the United States until 2006, when it was found in Idaho. The current hypothesis is that it was brought in on specialized seed tubers for production from a different location, probably out of Europe into Idaho. One last component of the story of the cyst spread is into Japan in the 1960s. The tradition in the 60s was the growth of potatoes in the highlands of Peru. They would bring it down to the coast in burlap bags and sell them. But they would reuse the bags at that point, and they would go off to the offshore islands and collect bird guano, bird excrete, high nitrogen, put that into burlap bags, ship it to Japan, and sell it as fertilizer, which was then put on potato fields. Unfortunately, the bags of burlap that contained guano also were contaminated with the cyst nematodes. And so the cyst nematode was therefore transferred with the guano from South America to potato fields in Japan. To summarize what we've covered so far on nematodes, they are microscopic aquatic animals. They live predominantly in the soil. Any environment that supports higher plants will support a specialized species of plant parasitic nematodes. In addition to parasitizing plants, there are free-living nematodes, there are mycophagous nematodes, bacteriophagous nematodes, entomophagous nematodes, and animal parasitic nematodes. Animal parasites are great examples of dog heartworm and Ascaris in pigs. Nematodes are associated with plants as parasites in predominantly 4 ways. As ectoparasites, where they feed at the surface of the roots and remain migratory throughout their life cycle. There are the migratory endoparasites, which invade the cortical tissues of the roots and the root tubers, etc., and feed internally, but remain migratory and worm-like throughout their life cycles. There are the sedentary forms, which once they start feeding, this worm-like structure is lost and they swell to become very globular, immobile forms. They may be completely embedded within the plant, or they may be exposed at the root surface. Hence, we refer to some as semi-endo and the others as endo-sedentary forms. Of the migratory endoparasites, the lesion nematode is a good example. A very simplified life structure, life cycle. Remains worm-like. All juvenile stages, the J2, the J3, J4, and the adults are capable of infecting the plant and hence can be inoculated. The potato cyst nematode and the soybean cyst nematodes are great examples of the semi-endo sedentary forms. Once they start feeding, they swell into these large globular females. At maturity, those females' cuticle becomes a leathery sac and protects all the eggs within it as a survival structure. Cyst nematodes tend to have limited host ranges, hence can be managed with crop rotation.